Guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will talk about protostomes and deuterostomes. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So in protostomes, mouth of the organisms forms before its anus during embryonic development. Whereas in deuterostomes, anus of the organism forms before its mouth, again during embryonic development. In protostomes, the developing embryo's first opening, that is called blastopore. So here we can see this is the first opening of the embryo. This is called blastopore. So this first opening or blastopore, it becomes mouth, while the anus is formed at a different site later. And here it is the digestive tube. Whereas the opposite happens in deuterostomes. So in deuterostomes, the blastopore becomes anus, the opening becomes anus, while the mouth is formed at different site later. In protostomes, cells divide with spiral cleavage. Here spiral cleavage occurs, whereas in deuterostomes, cells divide with radial cleavage. In protostomes, they show determinate cleavage, in which the development fate of each cell in the adult organism has already been determined. That means, suppose this is a 8-cell embryo and this cell is going to be a future heart cell, and this fate is already determined. Even if you isolate that cell from this embryo and keep it in another position of a different embryo, the cell will become part of the heart only. So that is determinate cleavage. Whereas deuterostomes show indeterminate cleavage, in which developmental fate of each cell in the adult organism has not been determined. So, suppose this is a 8-cell embryo. Now, these cells don't know what they will become in future. Suppose this cell will be future heart cell. Now you are isolating this cell and put it in the other location of a second embryo. Now the cell may become future brain cell or kidney cell or heart cell or liver cell. So its fate is indeterminate. Therefore, in this case, each cell of this embryo, if separated from this early stage embryo, remains capable of developing as complete organism. Each cell of this embryo is capable to form the complete organism. The coelom or body cavity, coelom or body cavity of protostome is developed by dividing the embryonic mesoderm. Hence, they are known as schizocillas. So, see this picture. This is the embryo. Here, this is the mesoderm. This is also the mesoderm. So, we can see that the mesoderm is splitting. Therefore, two mesoderm are here and they will become coelom. So, here coelom is there and the coelom is generated after the splitting of this mesoderm. Now, in case of deuterostomes, the coelom is developed by folds of archenteron. You can see this is the archenteron. Here it has two folds. This is one fold, this is another fold. So these two folds will make the coelom because these two folds will be pinched off to form that coelom in future. Hence they are known as enterocellars. So you know what is archenteron? So it is a primary gut, or you can say the digestive tube, which forms during gastrulation. Now the examples. So simple and lower organisms such as primitive invertebrates are examples of protostomes. And complex and higher organisms such as vertebrates are examples of deuterostomes.